we are starting our class. Here we are. Welcome back. It's our last class meeting before our spring break. I hope you have awesome plans for spring break. And so we're talking about Chapter 17 cell cycle. And as soon as we get back from spring break, we'll have finished Chapter 17. So we'll move on to Chapter 18, which is about programmed cell death. Apoptosis is a term for a type of programmed cell death. You probably heard of that. We mentioned it last time. And so we're going to get done with that. And then we want to start working on your term papers. And some people have started doing that. Spring break would be a good time to advance that some. Because, you know, when we get back from breaks, whether it's fall break or spring break, things happen pretty fast. And then the semester's done. Then we have an assignment to go over the exam 1. It's not going to change your score or grade. That's described in the answer key video, so you know where you are on that. We talked some in class at the start of class last time about you know, how to process results from exam 1 and build your learning from there as we move forward. So there's an assignment for that. It's due when we get back from break. And then here's a new assignment. So I'll post a video. It's a cell cycle drawing. We've done those before. It's a video. As you watch it, it describes drawing as you are learning, and so you know we're modeling that, continue to model the idea of draw it out, build it out, and pics of those drawings are due 2 weeks from today, and so that's where we're at for that. And today we're going to hear the third talk of 4 from Sam, and it's going to be focused on the T cell's response when it gets activated. And so it fits right into what we're talking about, cell responses and activation of those responses in this particular example. Take it away, Sam.
Thanks, Dave. Good morning, everybody. It's really hot in here.
Yeah, it's wonderfully hot because the science building is cold.
Okay, great. Today we're gonna talk. This is gonna be kind of my third talk about really about CAR T cells, and then we'll do a fourth talk about kind of what does this actually look like in hospitals and with patients. So today we're really going to talk about cellular response. I don't have my little flashcards today, so I'm going to be kind of doing this a lot to be like what comes next. So our first time here we talked about the idea that the way you work with proteins is you actually work with the genes. And we're going to create a chimeric gene, going to put it all together with a promoter that's normally expressed, and that will activate a variety of cell signal pathways. And what do those do? Those lead to a cellular response. So the first thing we're going to do, again, this brief recap of how does this work? We're going to take our T cells out of our patient. We're going to insert the new genes, the chimeric genes, into those T cells. We're going to multiply them and put them back into our patients. So we're never actually manipulating proteins themselves. But these CAR cells express novel chimeric proteins. Oh, that's funky. All right, looks like things are on top of each other. Okay, so quick recap. What is the CAR, right? It's a chimeric protein. It combines domains that exist naturally in nature but don't exist together. And we're putting them together by putting the coding regions of the genes that code for those different proteins together with a promoter that is normally expressed in T cells. So that way, this novel chimeric protein will be expressed in T cells.
Oh, Sam, so you had the PowerPoints in sort of this wide angle format.
Oh.
And I just dropped them in, and so we might see some of this.
That's okay.
And if it gets too frustrating, you can just leave.
Okay, sounds good.
I mean, if we could switch over to your actual PowerPoints if you want to. The only reason we're not doing that right now is just because it starts to mess up the recording, and I get all anxious about that. But let's just see how it works. I apologize that there's that overlap, and you'll probably see a little bit more of it.
Thanks, Dave. That's okay. We're all used to interpreting multiple layers of information on the same slide. Okay, just a quick reminder. The CAR, right, it's a receptor. It's going to bind to a cell surface protein on the cancer cells, and that's what's going to initiate it. And that's what's going to initiate this thing. So, binding, yay. Okay. The CAR integrates, again, we're still in, what did we talk about last time? It integrates multiple different signal pathways that normally are part of different proteins because we need to stop our T cells from killing us, but these T cells, we just want to go wild. So these different signal transduction pathways are all put together into one receptor molecule, one receptor that can activate all of these receptors, all of these pathways, excuse me. Okay. And then what do? They're going to have a response, right? The pathways get activated, and our chimeric T cells are going to do something. Before we go forward, do we have any questions about this, things that I need to clarify, things that don't make sense here? Okay, great. Cells we can roughly categorize cellular responses into two different buckets. What we can call genomic and non-genomic responses. A genomic response is basically anything that is mediated by changes in gene expression. So we're going to have genes that are being activated, transcribed into proteins, and become functional. These tend to be much slower. The body has to produce new proteins, has to produce novel proteins. Whereas a non-genomic response is a protein-mediated process that is usually much faster. And I think the really big number one takeaway from today's talk is that cellular responses are coherent and usually work together, right? And a great example that we love to talk about in biology classes is, right, stimulation of the sympathetic nervous system in a fight or flight response. You have, your body dumps epinephrine, which produces a bunch of systemic level changes in your body that all kind of make sense and work together. And that's going to be true with cells as well, right? The cells have responses that are coherent. So non-genomic responses, we're going to talk a little bit about these. I realize this is a little ambiguous, and so I apologize for that. So the non-genomic responses in T cells generally and in our CARs are the formation of something called the immunological synapse. We're going to talk a lot about that. This is probably one of the coolest things about T cells. So don't worry. So I know it's a little intense. And that happens through a rearrangement of the cytoskeleton. Cell death, really what I mean to say here is target cell death. Right? The T cells aren't going to die. They're actually going to proliferate. But the target cell is going to die. And the T cells kill them in two different ways. One of which we're going to talk a lot about in class, which is they induce targeted cell death, apoptosis, in their target cells. And they also release chemicals that directly kill the cell itself. And they release further chemicals to recruit chemicals called cytokines, which are just signaling molecules used by the immune system to recruit more immune cell responders to the area. Yes,
I have
Anna.
a question, and I don't know if you'll be able to answer it, but so we were talking about apoptosis, and that was, from my understanding, when the cell looks within itself and decides if it can go on and live or not. So how could another cell, how could another cell, because that would be like looking outside of itself, cause it to apoptosis?
Yeah. That is a good question. The short answer is that it takes a signaling molecule.
Oh, so it takes something into itself, and then it's like, okay, wait, I'm no bueno to itself. Okay.
Yeah, exactly. It's like.
So it has to get into it, though.
And it does that through a signal transduction pathway.
Okay, sweet. Thank you. Sorry.
Yeah. So it's kind of like we're passing so T cells are really these are the bad guys in a horror movie. T cells are like monsters, and they just kind of walk around, and they just pass people little notes. And most of the time, the little notes say hey, you're doing great. Keep it going. You're doing a good job. And then sometimes the notes say things like, die. And then you're a cell and you like get this note and you're like, oh my God, it's time to die. And then you do it. It's really metal. Okay, so this is the immunological synapse. And yeah, this is, so first we have an actual schematic of it happening on the left. And then here we have a little cartoon that I found that I think is really great because it's hug time, right? And so they're literally going to rearrange their cytoskeleton here to wrap around the target cell. And that is both going to increase the number of receptors on the T cell that get stimulated, right? So it's going to create a positive feedback loop inside of the T cell, leading to further engagement with this situation. And it's going to really specifically target these killer molecules, these little notes that say die, right, to make sure that they have a very specific effect within they're only killing the cells that they are targeted to kill. So this is a photo from the NIH, actually of T cells in mostly in green on the outside, 1, 2, 3, forming these, starting to wrap around and hug. And this is actually a cancer cell that the NIH has been able to visualize. And in red are the vesicles containing the cytotoxic granules, porphyrin, granzymes that are about to actually puncture holes in this cancer cell to kill it.
They actually make and release bleach, isn't it? Like hydroperoxide and bleach.
Yeah, I don't
It's
know a
crazy.
ton about that, but I believe it. Yeah. Okay, so here we go. Another schematic, right? The what I like about this one is it shows that this is receptor mediated, right? It is a receptor that is binding and inducing this action, right, through a signal transduction cascade. And it's releasing apoptotic factors and membrane disrupting molecules. And the change in the cytoskeleton causes it to release more and more and more of those as more and more receptors are triggered by wrapping around the target molecule. Genomic responses are, again, going to be coordinated. And what is the big takeaway here, right? These different signal transduction pathways that we've looked at are going to trigger transcription factors that are going to cause a coordinated effect within the cell. Broadly, we can think of that as T cell proliferation, T cell survival, and cytokine production are going to be the big three things, right? So this is a so our CAR T cells that have been taught to kill cancer are now going to make more of themselves, stay longer in the immune system, and generate even more killing capacity to target cancer cells. And this is a capacity that all T cells have. We are by combining these signal transduction pathways, are making use of these non-genomic responses, these genomic responses to create an even more effective cancer-killing cell. We're not going to dig into I know last time we went really deep in the weeds. We're not going to go really deep in the weeds on this one, partly because the current research on what these actually do and how these actually work is really up for grabs. And what's actually happening here with these different transcription factors is no one knows. So you can go get a PhD in every single one of these, just studying like each little circle on that board. The one thing I do want to say about this is that AP1, AP1/2, you see it in the second from the left right there. So here's a journal. This is 2019, Journal of Experimental Medicine. AP1 activity induced by co-stimulation is required for chromatin opening during T cell activation. Right, so the AP1 transcription factor is remodeling the chromatin, which is a mechanism and a thing we've studied and learned about. So I just want to kind of take this moment to say that these genomic responses are achieved through transcription factors like the ones that we have studied in class.
Awesome.
Great.
I forgot, do we clap here?
You can if you want to, Dave. You don't have to. Thanks, everyone. Does anyone have any questions about this, or thoughts, or curiosities, or things that I could explain better? Great. Thanks, Riley.
Awesome.
Thanks, Dave.
All right, thank you, Sam. And you can see that this example of just one, modern cancer treatment flows with our course. The topics to understand, well, what is happening in that type of cancer treatment? And people refer to it as cellular therapy. So we're not giving the patient a drug, chemotherapy, or radiation therapy. We're not doing surgery. Those are some other kinds of cancer treatment. This is a type of cellular therapy. So it's just really satisfying to see. And Sam will come in and talk another time about back to like, well, what is it kind of like from sort of the patient's perspective and in the hospital, just to kind of put that in context. It's a procedure that's happening in Boston. I'll give you an example of a friend who passed away from cancer and was preparing to go down to Boston and get that treatment. CAR T-cell therapy is still used as a last resort therapy. And it's because it's still considered somewhat experimental, and it's incredibly expensive. So it costs about $1 million, and they're only, have the capacity to provide it in scattered locations, like Boston, of course, and so forth. And my friend here in Maine got cancer, kind of lymphoma, a blood cancer, and so was getting treated, and they had some success, and she went into remission, and for several months, and then the cancer came back, and they tried, more treatment, and they were lining things up, preparing, if nothing else worked, to go down to Boston and have CAR T-cell therapy. But unfortunately, by the time it was the last resort, and to do it, her body was already too hurt to do it. So she decided not, they weren't going to go through that treatment, and she wasn't able to survive. Yeah, so it is, a really difficult situation that they're trying to, they try to deal with. But Sam will tell us that when patients have this treatment, they have a lot of success with this treatment. So a lot of recovery. He mentioned that the T cells will proliferate and stay alive longer, and they see that in patients that have had CAR T cell therapy. So not only do they stop the cancer, in its tracks very rapidly, but the patient keeps some of that protection in them for several years. So it's got a lot of promise, but again, it's tricky stuff. Yeah, Mallory.
Did you say that it was like a last resort treatment, obviously? Why? Is it because of the cost, or are there like other factors?
That combination of that it's still experimental, and also that it's only available in limited places, because it just takes, you have to take the patient's blood and take it over to a lab and really manipulate it in a lab. So there aren't very many hospital settings that are that sophisticated, and it has to be in a place like Boston or Baltimore, New York City, Los Angeles, San Francisco, these places where they really have like biomedical research hospitals all linked up to the patients like that. Yep. So anyway, other questions people have about that right now? Let's also check in then, back to our class, about questions people have in general about anything at all. You could bring up stuff from the exam that you wanted to ask about, you were confused about that you've seen so far. You can bring that up anytime, or anything that you've seen in the book, or anything about where we're at. Anybody have any questions? Yes?
I had a question about the kite. So we talked
About what?
About the kite.
Kit.
The kit, sorry.
Kit.
We talked about how it activates a bunch of pathways we've talked about, but then I have in my notes that it's one receptor that does two things. What did you mean, like it only activates two things, or all the pathway that it activates essentially leads to two options?
Yep, so the KIT receptor, and we brought that up. The KIT receptor is another example of receptor tyrosine kinase. So we learned about the PDGF receptor especially well, but we did learn that that FOXO story is also receptor tyrosine kinase, and we learned that when we activate the PDGF receptor under a scab, we really activate that RAS and then that MAP kinase pathway to stimulate new gene expression with ELK, transcription vector. And when we turned over to FOXO, we learned that that receptor pathway would activate the PI3 kinase, and so a different signal transduction pathway. And with the KIT, we actually activate both of those. And so it goes a little bit to being of a style like what Sam was talking about, where the CAR receptor activates multiple pathways. And so it's just a good example of that. It's a powerful receptor, and what it does is it combines the KIT receptor's signal transduction pathways, combine the topic for chapter 17. So through the RAS MAP kinase pathway, it activates the familiar cell cycle response, chapter 17, and then through the PI3 kinase pathway, it activates this decision to try to prevent this programmed cell death, which is chapter 18, our next topic. And you asked Sam a question about that, about apoptosis and that programmed cell death pathway, and we'll get to that in chapter 18. And so I mentioned last time that it's very typical that when a cell is stimulated to go through the cell cycle, it should start worrying. And what the KIT receptor does is it says, go through the cell cycle and try not to worry. So two signal transduction pathways, and like Sam, I mean, one of the most satisfying things about the CAR T cell therapy story is how the T cell makes a bunch of coordinated responses. When we combine those signal transduction pathways, we get several different things happening, and they all fit into what it should do. Like that? Yeah, so that's the situation with the KIT receptor. And some receptors have that, where they activate two pathways, some don't, like that. But so KIT is a good example of one that activates both those, and we're familiar with those pathways. The difference is that the PI3 kinase pathway, instead of activating FOXO in the KIT story, it activates these anti-death signals that we'll learn about after break. Yep. Other questions people have about anything at all? One time in my life, so never in my life did I go see my dad do his job. He was a lawyer, and he would go into court, and he would be like the kind of lawyer who cross-examines people. Never went to see him do that. That was sad and dumb, right? Should have gone to see him do that. One time, one of my children came in to see what I do, and she sat in the back, and she was about 11, and there was no option. She just had to come and sit in the back, right? All right, so it turns out she's very sweet, Lena, but she's a smartass too, right? So, I forgot to put it in context. Side note context is, when I was a child, we had lots and lots of pets, and one of the pets we had was a dog named Chopper. Random side note, it seems. But as we were walking out of class with my daughter, Lena, I was thinking about my dad and how I'd never seen him work. And here's Lena coming out, and I didn't ask her, you know. She just said, you know, okay, that was all right. And then I said, okay, well, what was it like for you? And she said, and again, she's a smart ass, wow, she said, you sure do talk about Chopper a lot in class? I was like, what? Right? And I don't remember why I thought of that. Who remembers why I thought of that? I don't remember why I thought of that. But it's like, I don't know what's going on. Maybe I'm having a little bit of a seizure. I don't know. But yeah, I'm shy, and I'm not really aware of what we're talking about. So yeah, I think we're doing well though. So we're foreshadowing, we're always foreshadowing, looking forward. And so simulate the cell cycle, simulate that programmed cell death. Something about KIT, something about Chopper. Chopper was a good dog. Beagle, purebred beagle. Okay, so here we are at the cell cycle, and we have these 3 points in the cell cycle that we are worried about. First one we're done talking about. It's the M phase checkpoint. It's the mitotic spindle assembly checkpoint. We talked about it. Today, we're going to talk about this one because it's easy. It's a nice package, and it's this G2 checkpoint. We really want to talk about start because that's the one where the cell cycle is usually regulated. It's just complicated. So see how we move around the circle. We physically move to the different points in the cell cycle, and we're not over there yet. We've been talking about it because that's where MIC works and other factors that simulate the cell cycle. They're going to be working over there. PDGF works over there. So we're going right there now. It's this G2 to M transition. And so we're going to talk in this chapter about these three topics. So we're there right now. Regulation of entry into mitosis, entry into M phase. And it's called the G2 to M transition, and it's a DNA damage checkpoint. If there's no DNA damage, the cell just rolls through, and the chromosomes condense, the nucleus breaks down, it goes into mitosis. But if there's DNA damage, we want to stop. It's like when the lid is up on the washing machine, and we don't want it to go into the spin cycle, because that's dangerous. It's dangerous to go into mitosis if there's DNA damage, because you're going to divide the cell before there's a chance to repair the DNA, and then you won't be able to fix the mutation. Okay? So this is we talked just briefly about this last time. This is the controller that controls entry into mitosis, and it's a kinase called CDK1. There are, CDK1 is a gene family of paralogs in our bodies and other eukaryotes, and they're given numbers. The only one we're going to learn the name of is this one. CDK1 works at the G2 to M transition. That's where it does its most important job. We'll meet the other ones because it turns out that CDKs also regulate start. It's just different numbers, and there are multiple numbers, and we're not going to worry about that. So learn this is CDK1, and then similarly, the cyclins are members of a gene family of paralogs, and they're given letters. So this is cyclin B. And again, over at start, we'll see other cyclins are involved, D and E and so forth. We don't have to worry about them because it becomes confusing. But in talking about G2 to M transition, we're kind of entering the introduction to talk about how start is regulated. Start is regulated by CDKs. So CDK1 and cyclin B, they're where we are right now, G2 to M. And the way it's going to go is if CDK1 gets activated, it's going to phosphorylate targets that are going to take us into M phase. So if it's inactive, the lid is up on the washing machine, and when it gets activated, the lid goes down. Cyclin-dependent kinase is what they're called. So you can think of cyclin B as trying to put the lid down. Cyclin B is like, you're good, go, okay? So that's what that looks like.
Did you say what the PDK stands for, or the PKF, or the M?
Oh yeah, let's talk about that for a minute. So when they're together, that's what it looks like when the cell is in M phase. When the cell is in M phase, throughout M phase, CDK1 is active. So it starts at the start of M phase, and it stops activity at the end of M phase. And so there's a name for that combination during M phase. The biochemist that discovered this called it MPF. So the active complex is called MPF. And that's kind of a detail, but it kind of acknowledges the people who discovered it, and it actually just stands for maturation promoting factor, MPF. And I mentioned last time that the scientists who really made the big discoveries in how the eukaryotic cell cycle was regulated were geneticists who studied yeast, and they looked at mutants to determine the genes, and they just called those all CDC with a number. And so that's just the way geneticists often do things, because they don't know really what it is. So they describe sort of their screening strategy for cell division cycle mutants, and then just number the genes, because they don't know what they are. On the other hand, biochemists usually are going after the biochemical function, and they'll name it based on what it does. So maturation promoting factors, and I mentioned that those were studied in female animals that were, when their eggs mature, the eggs are arrested, waiting in the cell cycle for fertilization. And that becomes a powerful way to ask, how are they stopped? And so the idea of a maturation promoting factor means that it stimulates kind of the maturation after fertilization of the egg cell. So that's MPF. Some people think it stands for mitosis promoting factor. If you want to call it that, that's fine, because it really is that. But it's really maturation promoting factor. Yeah, and our book calls it that. And again, I think it's just to kind of salute the people who discovered it, who are still hanging around talking about it. Here's a figure that's actually from our intro bio textbook that we use here, and it shows really nicely a little introduction to it. So there's what we call CDK1. There's the cyclin B, and there's the G2 to M transition, the G2 to M checkpoint, the DNA damage checkpoint. And you can see when they come together, we get MPF, like that. And that takes us into mitosis. And the reason we go into mitosis is just because CDK1 starts to phosphorylate targets. Okay? Next thing to notice is that this cyclin, CDK1, cyclin B, it kind of just builds up during S phase and then G2. It's there already. Entry into M phase is really not controlled by that cyclin. It's necessary, but there's something else that needs to happen. So that's going to be our focus. What else needs to happen to the CDK1 to activate it? Then you can see there's the MPF. It's active throughout M phase. And then when we finish M phase, we're going to turn off that MPF, turn off the CDK1. And that happens because the cyclin B gets destroyed. So our entry into M phase is different than our exit. Exit is caused by destruction of cyclin B. Entry is caused by something else. And that'll be our big focus. How do we get into M phase? Notice here, a big emphasis in introbio is that CDK1 is always present. It's in every one of our cells all the time. It's just inactive. And that's in general true for the CDK proteins. They're just there all the time. They're just regulated. They're off until they get activated. Okay? So what we want to do is, so that's important to appreciate, that in M phase, we've got that active CDK1. You can call it MPF, but the key thing is CDK1 is active, and it's phosphorylating targets. So we want to learn a little bit about the targets. What does it mean? Because it's literally the story of M phase is CDK1 is activated and phosphorylating targets. Okay? And it's always there, but it's only active during M phase. So our key thing is, how do we get that activated? Okay? And the answer is that, there's just the cyclin B going up and down, and the MPF, I guess, again. And then, this is a little bit out of place. Oh, this is just a side note to show you our book mentions some of the other numbers here. Don't worry about them. Don't worry about the other numbers. Remember CDK1, that's the only one I need to know. Other CDKs are doing the other star. Then cyclin B, we are learning that today. Other cyclins are over there at the other side. MYC activates a cyclin, not cyclin B. Okay? All right, so here's an image of our book showing the story of how to get the CDK1 activated. So there it is, activated. And how to get it activated? And the answer is there. And I will just zoom in on that. So I just zoomed in on that. So that's the figure from your book, and it just shows us how we activate it. And it's that this, although this is a kinase that's going to phosphorylate others, it's also the target of other kinases. And it gets phosphorylated on multiple different amino acids. Okay? So that's what's shown here, just the position and the amino acid abbreviation. Of course, you don't need to know that. Just the idea that it has a lot of regulation by phosphorylation. It gets phosphorylated by kinases. And the phosphorylation sites include a site that's needed for it to be active here, but then it also includes 2 inhibitory sites. So when those are phosphorylated, the CDK1 is inactive, and that's what's going to switch.
Can you say
If
that
there's,
again?
yeah, I'll say it that again, but I'll say it a slightly different way. If there's DNA damage, the cell cycle stops at the G to M transition because this CDK1 is phosphorylated at those 2 inhibitory sites, and it's inactive. Even though there's plenty of cyclin B, it's stuck. And when the DNA damage gets repaired, those 2 phosphates get removed. So now we'd say, what is the thing that removes the phosphates? And the answer is, so far in our course, we've talked about kinases adding phosphates and how we have 600 different kinases in us because we do that a lot in a lot of different target settings. Oop, why is this not working yet? Oh, we have a little bit of an issue here with my, oh, so let me just swap out here and get this fixed up. Sorry, it's just my Bluetooth is not doing its thing quite yet, I guess. Come on. Trying. Oh, let's remove some of these others. Sorry. Yay, okay, sorry about that. I was delaying the game there. Looks like our recording's still going now. It's a good thing. All right, so we zoomed in on this, and we were just talking about how there is a kinase, so we can just draw this out, because kinases are enzymes. They must catalyze a chemical reaction, and they catalyze specific chemical reactions. So we're going to have a kinase that has CDK1 as a substrate, and it's going to phosphorylate that CDK1. Right, so that's what kinases are, and each one has specific target molecules that it's phosphorylating, like the PI3 kinase phosphorylates phosphatidylinositol, and so forth. So we have a bunch. So there are some kinases, and we don't need to learn the names of those in this example, but what we do need to learn that's new is, hey, what if we need to remove that phosphate? And there's a whole different set of enzymes that remove phosphates, and those are called phosphatases. And we're only going to learn the names of a couple of them in our course, maybe 2 total, but they must exist, right? Because you want to remove those and turn it off. And the one we're going to learn about is the one that removes these inhibitory phosphates, and it removes both of them. And it is called CDC25. And it's the one that removes it. And so that's who's really controlling the entry into M phase. And we'll see that CDC25 gets turned off when there's DNA damage. So we're looking at a very simple little switch system. We just have to detect DNA damage, then inactivate CDC25, the inhibitory phosphates remain on CDK1, the lid stays open on the washer, we get stuck at the G2 to M transition until DNA damage is repaired, CDC25 is activated, it removes the inhibitory phosphates, CDK1 gets activated, we have the MPF ready to go, and the lid goes down and we roll into mitosis. And CDC25 is just named from the geneticist that first discovered it in Seattle. Okay? So there it is right there. It's just pointing it out here. It's the transition is for CDC25 phosphatase to remove those two inhibitory phosphates. And if we go back, you might be able to see from a distance that we go into mitosis, CDK1 is active because we remove the inhibitory phosphates. And it's two, no big deal, it's just two, like that. The activating one is still present. And so we're going to roll in to M phase. We're going to be in M phase because we have the activating phosphate and we've removed the inhibitory phosphates. Then we're going to complete M phase and destroy cyclin B to leave M phase. Yep.
Who adds the activating phosphate? Is that?
We don't need to know,
Okay.
but it's listed here. Yeah, we want. Another geneticist name, right? Because if you're mutant for we want, the cells are really little. Yeah, yep. So not a big deal, because what we want to focus on, the big deal, adding CDC25 to the story. Then we're almost done with this important switch, because we just need to look at what's upstream of CDC25, what's regulating it. And it turns out that CDC25 has a very simple upstream regulation, and it looks like this. Unfortunately, our book shows this as a complicated figure, and I'm just going to circle part of it that's relevant for us right now. And that is DNA damage is present, a kinase called CHEK1 gets activated and adds an inhibitory phosphate to CDC25. And I apologize because now we've got a lot of different kinases. What? You know, a cascade of kinases. That's confusing. But CHEK1 is a kinase, and when it is DNA damage has occurred, it is going to add a phosphate to CDC25 and stop it. And that's it. So we really have, and you can see there's a little bit of action up here in detecting the DNA damage, but it's a very simple biochemical pathway to control the entry into M phase, because the entry into M phase, again, is just a DNA damage checkpoint. So it is a simple switch. We're only worried about DNA damage. There's plenty of cyclin B. We're good to roll. But what if there's some DNA damage? We don't want to go. So it's just a, it's just a very little, nice little biochemical check like that. And it's satisfying, right? Because we just learned all of it, right? We could dive into the weeds of saying, well, who adds those phosphates in the first place? We don't want to go there. We could talk in more detail about the DNA damage and the type of DNA damage and how that's detected. But we don't need to go there because we have a nice little thing. DNA damage activates CHEK1 kinase. CHEK1 kinase phosphorylates CDC25 and stops it. CDC25 stays stopped until the DNA damage is corrected, the phosphate's removed, and then CDC25 removes this inhibitory phosphates from CDK1, and we're in mitosis. That's the switch, total switch. Nice. And that's why we went there. It's because it's a nice, satisfying switch with cyclin and CDK1, cyclin B and CDK1. We're done with that. The only reason we're going to go back there is at some point we'll mention what are a couple of the targets in M phase, because the book talks about them and the targets are super satisfying for us to learn. But for today, it's enough just to know the switch, the control of the switch, and that's it. Yep. So any questions about that? And you know, you can loop back for questions anytime, or forward or sideways, you know, and my job is to fit them in, you know. So, you know, if you're thinking about a question, it's distracting. Like you might ask, was Chopper a purebred beagle? Yes. Yes. Did Chopper have a registered name? Yes. Was it Danny's little Chopper, Grumper, Zeezer, Pupster, Champlin? Yes, it was. Yeah, very good dog. And yeah, apparently I talked to Chopper subconsciously to this day, right? So regulation of start. It's a story about activating a CDK, but it's a different CDK, and we're not going to worry about the numbers. And one way it gets activated is cyclin-dependent kinase. It needs a cyclin. What happens under our scab is the PDGF goes through all that pathway, activates MYC, and MYC simulates the synthesis of a cyclin. Not cyclin B, different cyclin. We don't need to worry about the letter, but a cyclin. And that's the rate limiting step over there. So, in the G2 to M transition, is cyclin B the rate limiting step? Nope. The focus is on those phosphates. How about over at start? That place we're really interested. What limits the CDKs at start? Sometimes it's cyclin. You just need to make cyclin and you're good to go. Okay? So see, we're going to add more when we go over there. It's a cyclin CDK story, but different CDKs, different cyclins, different point in the cell cycle, and it gets more complicated. So I don't think our book has this figure anymore, and it's too bad, because this is the story at start. At the start, in the G1 to S transition that we're really focused on, that is typically controlled. And it looks like this. There's a CDK, not CDK1. Don't worry about it. Just there's a CDK. It's going to phosphorylate targets to go into S phase. There's a cyclin. We don't need to know the letters because there are multiple letters. It can be complicated. This gets really complicated fast. But this is a nice picture. Just a CDK1. That's going to be the action player. It needs cyclin, cyclin-dependent kinase. Sometimes the cyclin's the rate limiting step. There are phosphate stories here, and those are not something we're going to worry about. So we can kind of keep it simple, right? We know there are big time phosphate stories of activating and inhibiting the CDKs at start, but we won't dive into them in this course because we have a great example there with cyclins. Sometimes they're really linked. And then we want to introduce another one, and that's this. And there's a bet, I don't know anyone that calls this that. Instead, people refer to this as small inhibitory proteins. So that yellow thing can be one of several little proteins that are generally referred to as small inhibitory proteins. You know, for exam 2, what's the big difference between CDK regulation at G2 to M versus G1 to S? At G2 to M, it's all about phosphorylation. At G1 to S, yes, there's a phosphorylation story that we're not going to learn about, but it's there, plus cyclins, plus small inhibitory proteins. It gets more complicated. And we'll learn a couple of examples of small inhibitory proteins. Small inhibitory proteins is a great name because they inhibit. We've already used the analogy of a green light, red light, and these can just make the light stay red. Or, even if it turns green, they can block it. They kind of can outclass anything. Just don't go. So these are just like a warning, right? They must be warning the cell, just don't go into S phase. Yep. Remember that cancer is uncontrolled cell proliferation, and cancer is going to be causing trouble in this mechanism. Like that, okay? Oh, yeah. So, examples of CDK regulation at start that we'll focus on. Cyclins, phosphorylation, and binding of inhibitory proteins. And these are just examples of two we'll learn about, P21 and P27. And we'll just go through them one at a time. Just to introduce the small inhibitory proteins, here's just a gel, electrophoresis gel of those two proteins. And it's a polyacrylamide gel. They're just looking at proteins. They have purified proteins. And biochemists give good names. Sometimes they give names where they don't know yet what the protein does. So they just call it a P for protein and its size. So the size of proteins are measured in an atomic unit called Daltons, and P21 is 21,000 Daltons, or it has a molecular weight of 21,000. So that's why they got those names. Yep?
Why is it next to the 46 kilodalton?
That's a good thing. I didn't even look at that because I just grabbed this off the web. And yeah, that somehow got shifted or was wrong. That's a good question, because yeah, it's just in the wrong place relative to the molecular weight standard. Yeah, good call. Huh, yeah, I don't know. I didn't notice that when I grabbed it. Yeah, because shouldn't be running up here. It should be way down there. Yeah, but yeah, so it's out of place there, so not a perfect example. But a key thing is just, why these simple names? P21, this stands for a protein of 21,000 molecular weight. Yep. Okay. And our book lists these here. See, it's got P15, P16, P18, P21, P27, like that. So there are a bunch of them. And do we learn all of them? Nope. We learned two of them, P21 and P27. And we'll have examples of them, like that. You'll be able to go, that's what P21 is doing, that's what P27 is doing. Do they play a role at the G to M transition? No. That's all about the phosphorylation, right? They're over at the start, and they're inhibiting start. And it's because we need to be really careful there. That's the big control point. And so this evolved as an extra layer of protection. And it's good that they're inhibitory. They're just going to be able to stop it. All right? So we're going to talk about 5 examples of regulation of start. So it's regulating the CDKs that control the red light, green light at the G1 to M transition. And these are the 5. So you see P21, P27 come up, along with another P, but it's not a small inhibitory protein, it's a different protein, it's a big one, and other things. So the first thing is, well, we already talked about this, because we talked about genomic response where new gene expression occurs under a scab. PDGF stimulates synthesis of MYC. And MYC is a transcription factor that activates cyclins. And cyclins are often rate limiting. The enzyme CDK is cyclin dependent kinase, and there just aren't enough cyclins. So it's often the case where the light is red just because we're waiting on the cyclins. And if some transcription factor can activate them, they'll go. And so far we learned the name of one, MYC. And we learned that MYC is a dangerous gene because the body tries to not make very much of it. And if you make too much, you'll get cancer. And so it's an oncogene. It can get disrupted by just making too much of the normal MYC. So we already know a fair amount of that first example. But we're just going to add a little bit to it now because there are other stories there as well as the PDGF and the MYC story. So here's that MYC story from exam 1, right? PDGF receptor pathway under a scab. MYC gets activated. Yes, MYC, specifically the cellular MYC, the normal MYC is activated in skin cells during wound healing. Its activation is particularly noticeable in the transition of cells from G1 to F. Great, right? So we know that, okay? And we also know here is the target of elk. So we're going to dive into that and expand on that a little bit more because it's our good example. And then we'll have a couple other things come up too, okay? So here's just one thing. Some people did CHIP analysis, chromatin immunoprecipitation analysis, to identify all the targets of MYC. And MYC has a lot of targets, and they all kind of do the same concerted thing, including activating CDK genes. So just know that MYC is doing that, and it activates other genes. We see that really typically, that transcription factors have multiple target genes, and they do coordinated things. It activates a couple of different cyclins. And see, cyclins have letter names, and we're just not going to go there for these because it's several, and they get complicated pretty fast. Yep. Okay. And so MYC responsive genes, right? Like that. So, oh, this is a little bit out of line. So let's just zoom in. We saw this figure. This is our figure. I'll just prove it. I'll just scroll back here. This is our figure. And we're going to zoom into the MYC cis regulatory region. And I'm just zooming in on that image. And we see elk. Elk got phosphorylated by ERK, and there's elk. Go for it, elk. Yay, elk. It binds to an enhancer, and now we're going to learn a little bit more about this situation. Because the enhancer is called SRE. So we're going to learn what that stands for, because again, we're in this first example of the regulation of start and making more cyclin. So this is a cyclin gene. Meow, 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 meow. So there's a cyclin gene, and you know, I don't label these in detail anymore, but you know what they are, and that's just a cyclin gene. Okay? And if we activate its expression, we'll have plenty of cyclin protein, and we will turn the light green and go into S phase. So here's the enhancer of this one, and this region, this enhancer is referred to as a SRE, and it stands for serum response element. And elk's going to bind there. So that's elk. And now we're going to learn there's another transcription factor that binds to the same enhancer and is also helping. Yep.
Is this the MYC gene or the cyclin?
Oh, you're right. It's MYC that we're focused on. Yep. Yes, thank you. So this transcription factor is called SRF, and it's called serum response factor. So these guys team up to activate cyclin and drive into the cell cycle. And they bind to this thing that was originally called a serum response element. And that's because there's a story that goes like this. And let me just tell this story, and then we'll have a little concept chat. So, where we go here. So, in research labs, scientists actually often grow cells in dishes with nutrients. That allows them to do a lot of research on humans because it's not a human being, it's human cells. And they're growing in a dish, and you can do a lot of experiments on them. And the dish includes nutrients like amino acids and sugars and vitamins and minerals, everything the cell needs. But it also has added sort of a magical potion that the cells need to grow. And it's surprising to hear this for the first time, but when they add the nutrients, they add 10% of what's called fetal calf serum. So an unborn calf is, they collect its blood, and whole blood is made of liquid and cells. And if you centrifuge whole blood, whether it's a calf or a human, centrifuge whole blood, the cells pellet, and the liquid is called serum. And that's where growth factors are. And so this fetal calf serum that has to be added is they're adding these kind of magical potent X factors that are growth factors to stimulate the cells to grow. And the people have been growing cells like this for years and years and years. There's a famous example of a woman from the 1950s named Henrietta Leakes, whose cells were taken from her body and cancer was removed, and the scientists started growing the cells in the dish, and they just kept growing them because her cancer cells could live forever in the dish. And they didn't tell her family, didn't tell her, and they just grew them like that. And they're called HeLa cells after her, but their family didn't know. And they're just growing all over the world now. So that can happen, but they need not just the simple nutrients, they need some growth stimulation. So this is where we get the serum response element and the serum response factor. Because what the scientists would do back in the 1950s, they'd be growing these cells and they'd just remove the serum. The cells were happy, they had all the nutrients they needed, but they would stop growing. And where they would stop is at start, because they didn't have a growth factor to turn the light green. And then the scientists would add the growth factor, add the fetal calf serum, and they would start to grow. And they would all be synchronized, so they could do experiments on them. All of them would be stopped, and all the cells would go forward together. And they saw new gene expression. The fetal calf serum caused a genomic response, new gene expression. And they isolated those genes. They didn't know what those genes were doing, but they said, let's try to identify the enhancer. And we talked about how they identified the enhancer. And the enhancer must be a serum response element. They somehow activate that new gene expression by being activated by the something in the serum. And so let's find that transcription factor. We'll call it a serum response factor, like that, right? And so it's a very simple story and a very old story about regulation of the cell cycle. Even before they understood that there were growth factors in the serum, they knew the serum was doing something special, and if they removed it, the cells would stop. If they added, the cells would grow. They didn't even know much about it, but they started going after that. And so we just want to add that detail to this story. That, yep, transcription factors regulate cyclin. So we're going to regulate the production of MYC or cyclin, like that. Okay, so that figure has always had that, but you know, it's too much for our first exam, but now we're just adding some to it because we're really interested in START. So here's just a little bit about what's called the restriction point, and it's called the restriction point. START, another word for START that's synonymous with START or the G1 to S transition, is the restriction point, because the cells are growing and then there's some restriction. We're like, we can't get through that. We need something. And the restriction point, the point at G1 at which commitment occurs and the cell no longer requires growth factors to complete the cell cycle, has been returned the restriction point. So it stopped there, and if you add the growth factors, it'll go forward. Yep. And it's phrased there a little bit kind of backwards, the way they express it. It's kind of just a backwards sort of way of stating that situation. Great. And it's shown here in our book, where this is the RAS, ERK pathway, activate L, and expression, activation of a transcription factor, activation of cyclin, and we go through what's called the restriction point there. It's START, it's G1 to S transition. Just another way of getting at it. In this case, they don't mention MYC, because there's a different story with different transcription factors that's used in the cells they were studying. They weren't studying skin cells under a scab, they were studying some different cells. And in those cells, it was the same signal transduction pathway leading to the same effect, activation of cyclins, but it was just a different transcription factor, and it's this one, and it's made up of two proteins, so it's a heterodimer that binds not to activate MYC, but to activate the cyclin. So it does the same job as MYC with the same pathway, it's just different cells than the skin. So things get complicated, right? And they're called June and Foss, and they're, like MYC, famous oncogenes. Because if they get activated, you're gonna go into the S phase like that. And together they're called AP1, and Sam mentioned June, and that was on his slide. Those are transcription factors that get activated in a T cell, like that. And this doesn't show up until chapter 19, which is more about cancer. And so that's where the book kind of spreads this story out a little bit. And so let's pause for a little concept chat. See, it's START, and START gets complicated, and we're adding carefully some pieces, right? We got a list of 5 things we're going to go through. The first thing is the story of transcription factors regulating cyclin synthesis. We already know some, and we're just going to add a little bit more because there's a lot more going on. For example, the CAR T cells, they get stimulated to go through the cell cycle by the RAS, RAF, MEK, ERK pathway, but not MYC. This June phos and the complex is referred to as AP1 because they work together. Like that, okay? So you can start thinking that here's the cyclin gene, and it has an enhancer that can bind June phos, and probably also has an enhancer that can bind MYC. And either of those could get the job done. So we'll actually draw that out. There are multiple cyclin genes, so maybe it's not one, but we can pretend that we can have a model of how many enhancers might it have. So far we know 2, MYC and June phos. Yep, so let's do a concept chat. We have time for one, and then we'll blow out of here on spring break. Party all the way to Puerto Rico, you guys, it's gonna be great. Try to talk with somebody new, and you can ask them, what are you doing for spring break?
Spring break I'm working, and I'm gonna finish my well, I started it, but like I'm still just gathering information.
It's called DR4. It's called the death receptor.
So the fetal capture amount really interesting me to learn about because I was at lab like all the time with my cells growing up, and it is so expensive. But I guess I didn't know why we needed it. Like I just knew we needed it, but that's really interesting to know. It is like unimaginably expensive.
I know. I use it. I just spent 15 bucks. And I was like, we had like a freezer full of it. And I was like,
We had this guy that was here lots of times, and he said he works for the software company that does that, and he said it's really common for them to get orders of over $1 million just that stuff. Like, holy things.
so I was confused originally about cyclin 2 because I thought cyclin was for G1S phase, not for G2N phase. But then I learned that there's 2 different, well, there's a lot of different cyclins, but specifically cyclin B and just cyclin.
Yep. And then I learned that our for the this is the first one still that it has to be dephosphorylated. So this one's going to be activated by dephosphorylation. Well, there is phosphorylation in one thing, but we'll talk more about that.
That's not.
Yeah. So the CDC25 will remove its phosphates if there's no DNA damage, and it'll turn off if there is damage. So then it's like phosphorylate. If it's phosphorylated, it can't be shot.
Once the cyclin on MP, once that get phosphorylated, is that what makes it go
You lost me. Okay, so the CDK1 is up here.
So you know what? Yeah. So when it combines together to make NPF, and it is a cyclin-dependent kinase. Is that like a phosphorylate cyclin? Is that what makes it?
I think that this is the blow up of that.
So we'll do this for another minute.
I believe that this is showing us more of this. So this CDK1 has to bind to the cyclin B, but then that doesn't even activate it. It's the phosphorylation it's the dephosphorylation.
That's what originally binds it. After it goes through
No, it can't go through unreads until after that. Until after it goes through.
But you know how it degrades after this.
And Mariah, what's one thing that your group thought was important so far today?
We talked about the small inhibitory proteins being part of the G1S regulation, but it's not the G1.
Awesome. Yep. right? Yeah, so we very intentionally went to G2 to M to get a handle on that and to appreciate how satisfying the mechanism is. The mechanisms over at START are satisfying like that. Also, it's just complicated because we have a very complicated, dangerous situation over there that has multiple different angles, you know, like that. Emma, what's something your group talked about?
We talked about CDC 25 and the importance of that when it comes to taking the phosphate group off of CDK1.
Yep, awesome. CDC 25, I said that was discovered in Seattle. I went to grad school at Cornell in Ithaca, New York, so on the East Coast, and my first year as a grad student, there was a second year grad student who was so beautiful, I would never talk with her. I knew I wouldn't be talking with her, and I didn't intend to. And I swear to God, I did not intend to. But by the end of my first year in grad school, she and I drove cross country to visit that lab in Seattle. Like, that's how weird things got really fast. Yeah, you know, and I'm like, yeah, let's go. And we should probably go to a couple of Grateful Dead concerts on the way and meet some of her family members. And she's like, yeah, but let's make sure we get to Seattle. Yeah, it was super weird. And I was just like, yep, yeah, oh God, I don't know what's going on. Yep. Yeah. Side note, some of you already heard in lab classes, on our first date, our first date was stimulated by her coming in to lab late at night saying, I want to go with her and try to dig up a human grave. So, anyway, CDC 25, you know, and it's studied in yeast. The place is called the Fred Hutchinson Cancer Research Center, you know, in Seattle. And they had this guy studying yeast, you know. What a rando, right? You're like, what? The yeast that causes bread to rise? Seriously? At a cancer center? You know? And and we had to go there just like as fast as we could go. Yep, okay, and let's see, Matt, what's one thing you guys talked about too?
Well, we're talking about the cyclin B because I was confused at first when we talking about cyclin. I thought it was from the G1 to S phase that there was cyclin, but there's more than, obviously there's a ton of different cyclins, but specifically cyclin B for the G2 to M phase. Yeah,
and we're gonna try to keep things sorted out by just learning the name of the ones that regulate G2 to M, cyclin B, and CDK1. That's where they do their job, their big job. And we'll just call the other ones CDKs and cyclins. And we won't worry about that MYC is activating cyclin D2 or anything like that. It's a cyclin. That'll be great. Yep. And I'm glad that nobody mentioned Chopper. And I'm sorry that nobody mentioned Sam's talk, because that's good stuff. Students really love cellular biology. You know, when the cell is doing stuff like, you know, it's just gonna hug that cancer cell, you know, because it's gonna be feeding at these death factors, and it's gonna be, like Sam said, a positive feedback loop. So that's our spring break. That's our spring break vision. Two things. One is, remember to, if you work on your term paper, anytime you work on it, work in those documents. That's a big key part of it. And the other thing I wanted to mention is just this. When you learn stuff, there can be this aha moment, and it's a natural high, right? Endorphins get released because you're like, whoa, I got that, right? And there's a lot of opportunity for that here, right? Because we have a lot of little pieces that seem complicated, and then you're like, got it, I got it, you know? And so I hope you have some of that during spring break, is some natural highs. Learning highs. Go for the learning highs on spring break. Come on, you guys. Party with your textbooks. Just do it. All right, yes, and we'll be